Okay, next up we've got RNA processing. And so eukaryotic cells will have three types of post-transcriptional modifications within the nucleus. Keep in mind that this is in eukaryotic cells, not prokaryotic cells. So in eukaryotes, we've got introns being removed and exons being spliced or joined together. What are introns? I think of them as interrupting regions of a gene. So they are non-coding regions, whereas exons are coding regions. And remember that exons are expressed. So exons express, introns interrupting. This means that mature mRNA is shorter than pre-mRNA because the introns will be sliced out. And I think I've got a diagram on the next slide where I'll show you this. You've also got a methylguanosine cap being added to the 5' end of the RNA molecule and a poly A tail being added to the 3' end of the RNA molecule. And once these modifications have taken place, the RNA molecule will become mature mRNA and it will leave the nucleus to move to a ribosome. So you can see here we've got that pre-mRNA, we'll have the introns being spliced out and the exons, which are expressed, joined together. We'll have the addition of a 5' cap, methylguanosine cap, and a poly A tail, which pretty much just means a long chain of A's added to the end of that. And these kind of assist in export from the nucleus. Same diagram once again, just kind of showing it a different visual. So why do we remove introns? So it's really important that we remove introns because these regions do not code for protein molecules and they will be useless in translation. We do use these regions in some genome modification processes, but we don't really discuss this in VCE. So don't worry too much about it, unless you want to. So one gene may be able to code for more than one protein or variation of a protein depending on which exons are kept in or left out, and you'll have alternate exon splicing. So you can actually shuffle about these genes. You'll look at that if you do biomed in uni. I really enjoyed that and I thought it was super interesting. But yeah, we don't really look at that too much in VCO or VCE. This may also help explain why the amount of proteins in the body are so diverse despite the amount of DNA present. So we've got the same DNA in all of our cells, but different cells will express different proteins. So our skin cells will express particular proteins needed in the skin, whereas our liver cells will express different proteins. Our kidney cells will express different proteins. They all express different proteins. And that's because they kind of shuffle about the exons and have alternate splicing. So what is the point of that 5' methylguanosine cap? It's really important because it helps to initiate the process of translation, which is recognition at the ribosome. Both the 5' cap and the 3' poly A tail protect the RNA strand from damage from enzymes, and they also add stability. The 3' poly A tail also assists in the export from the nucleus. So that mRNA is going to leave the nucleus via a nuclear pore, and it's going to go off and travel to the ribosome.